All right, so the way I tend to talk about the cell membrane is the fluid mosaic model. There are a variety of ways of conceptualizing the cell membrane. That's the one I tend to use. And in that model, it's a little bit like the lipid bilayer is a fluid membrane. And then floating around in that fluid are kind of like these icebergs of channel proteins and membrane-associated proteins, right? So here's our lipid bilayer. The little yellow guys in there, that's cholesterol. Okay, remember I said cholesterol is actually a requirement for our physiology. It keeps the membrane nice and fluid. Sticking up off the membrane are proteins that are called glycolipids, and together they form what's called the glycocalyx, which is kind of like a forest of little molecular trees sticking up off the membrane. These molecules play a variety of roles. You're going to talk about some of those roles in immunology. They help cells to identify each other. They help cells to interact. Okay. Here, right in the middle here, this is a channel protein. This channel is probably open all the time, right? Because you don't see any mechanism for it to be closed. Here's another kind of channel. This is a gated channel that can be either open or closed. And then we have structural proteins, like this little helix here has a component outside the cell and inside the cell. And these proteins that are structural, they help to give the cell its shape and allow it to like connect to other cells and form a tissue. So this is mostly I put this slide up here so you can kind of conceptualize. You've got this lipid bilayer, and then you have these complicated shapes, these protein shapes floating around in it. And how the proteins change their shape is going to create their function. Okay, so for example, so here's our lipid bilayer, right? Here we've got a couple of different examples of channel proteins, right? So here's one that's allowing simple diffusion, right? You can have another that's allowing facilitated diffusion. In other words, there's not really a hole there, but the protein can change its shape to make molecules go according to their concentration gradient. And then in terms of active transport, we use energy to move a molecule against its concentration gradient, so towards the high concentration side, right? So I like this example of facilitated diffusion because it kind of shows you how these molecules can slow things down, right? So here's the molecule to be transported. Let's say it's glucose. It comes in from the outside, so extracellular fluid. It binds to its binding point. And when it binds, it causes a molecular change, which actually changes the shape of the protein. That change in shape makes the transported molecule makes it bind less well here. So it releases and goes inside. And then that causes the protein to go back to this shape, and that process repeats over and over again. So it's like a machine that takes a molecule in. When it connects, that connection makes it change. The molecule is released. Releasing the molecule makes it change back, and then it does that over and over again, kind of pivots. And then over here on this last side, this is a gated channel. We're going to talk a lot about these this week. Okay, the sodium and potassium gated channels are what create the action potential. And the action potential is basically a way for the cell to change in one place and very quickly change in all other places at the same time, or at least in a very short amount of time. So here for our gate, you can see that this protein has a strong negative charge inside, right? Even though it has that strong negative charge, when the gate's closed, sodium still can't get in and make anything happen. But when a change occurs to this protein that allows the gate to open, now sodium is suddenly attracted to this negatively charged inside, and it kind of gets pushed into the cell. So it goes down its concentration gradient relatively quickly. The same thing happens for potassium in reverse. Remember, potassium's high inside, low outside. So when the potassium channel gate opens, the potassium very quickly exits the cell. Now, the key to that is not just that we're moving sodium and potassium, but that we're also moving a little bit of charge too, right? Because for every sodium ion that moves, we didn't just move a sodium, we moved a positive charge, right? So we're going to use that when we go to build membrane potential here.